in the chat if you can answer. Yeah, thanks a lot.
Sure, I'll put in the chat.
Alessandro Nicoli. Alessandro is part of molecular modeling group at University of Munich, together with Antonella Di Pizio, and he will tell us about sequence and structural information of serotonin receptors to guide the ligand discovery of TAR receptors.
Okay.
Alessandro, take it away.
Okay, could you hear me?
Yes.
Okay, perfect. Thank you everybody for being here, and thank you, Katarina, for the nice introduction. And today I'm really happy to share our computational work on ligand discovery of olfactory receptors. They are the most numerous GPCRs, and they comprise two groups. One is the 400 odorant receptors and 6 trace amino-associated receptors, and they're expressed in the nose. Mainly, they mediate the perception of smell, but interestingly, they are also expressed outside the olfactory epithelium, where they are involved in other physiological function or pathological condition, and they might represent interesting drug target. But today we will focus our attention on the TAR family. They are basically receptors that recognize small volatile amines, and among them, the TAR1 is the most well-known, characterized, and also as a drug target for neurological disorder. And we can see here below two of its two ligands. But TAR2 and TAR9 are considered orphan receptor. Indeed, we need computational protocols to discover novel ligands that they might serve as a pharmacological tool to shed light on their function, or to put the basis for a drug design campaign. When we start our journey, we choose to work with the mouse TAR5 since we had some ligand in the literature. There were two agonists and two antagonists. And then talking with our collaborators, Professor Stefano Espinosa in Genova, we run an high-throughput screening, and we enrich our pool of antagonists with new 4 antagonists. And then here you can see the chemical structures. They have low micromolar activity, but also is really interesting because these known antagonists for TAR ligands are also, they share a pharmacology with already serotonin receptors. Again, starting this work, we didn't have any structures of any TARs, and basically we generate our homology modeling based on the beta-adrenergic receptor 1 and 2 in their inactive state, since they share in the full length the highest sequence identity. But when zooming into the binding site, we can clearly see that the serotonin receptors share an highest sequence identity locally. And also is interesting because some authors propose that the TAR gene are also basically originated from the duplication of the serotonergic receptor 4. Then, since these two classes of receptors, they share similar ligands, it means that some conserved residues, they might form similar interaction, and also they might have a similar poses. Then, also for that, at the time, we didn't have any experimental structure of this serotonin receptor. We collect data, structural data from GPCR-DB, Rosetta GPCR, and Swiss model, and then, guided by the docking and literature data, we generate multiple docking with for each ligand with its cognate receptor, and here we can see 2 examples. But then, interesting, when once the structure has been released, we also achieve a good sampling at the time with our refined model with the receptor 1E. Then, taking together all this information, basically from the sequence, we learn which are the conserved position that we want that are will have a rotameric state similar to the known TARs or other serotonin receptors, but also some TAR5 specific position for which we want to sample their conformational space. And from the structure-based comparisons with the serotonin receptor and the fingerprint analysis, we get to know which are the important interaction that we want to have in our docking. Therefore, we select 11 residues for which we sample and we explore the conformational space inside the binding site. How our protocol works, as you can see here in the video below, basically we generate many conformation using induced fit docking. And then each of these 2000 conformation that we generate has been evaluated with an automatic pipeline for which it gives you back the docking and also its RO curve. We select 2 models with an improved RO curve, and then these 2 models has been used for a virtual screening campaign for which we use a specs library. Then to reduce the number of molecules to give to the docking, we use classical molecular descriptor or filtering. We use a pharmacophore-based filtering, and then using both separately model, we perform molecular docking, and through visual inspection, and then a part of the ligands using interaction fingerprints, we came out with 29 compounds that has been experimentally tested in Genoa, and surprisingly, we discovered some drug-like antagonists also for our TAR5, and with a hit rate of the 10%. Then, during the time of this journey of this project, also AlphaFold came out in 21, and we compare our refined models. And if we can have a look here on the picture on the left, we see that our refined models pose the sum of the conserved residues in a similar way for which also AlphaFold that has been trained with some of the serotonin instructors in a similar position. Then also in 23 last year came out also the first structure of the TARS, in this specific case the mouse TAR9, and also here we can see that some of our similar residues of our refined models pose the side chains in a similar way and orientation. Then we also wanted to explore how stable were the poses of the novel antagonists from molecular dynamics simulation, and we can see here a small video of compound 9, for which we can see that during the 200 nanoseconds of simulation for 3 replicates, it stayed quite stable. And then we also, since these poses were stable, we also compute the interaction of these 3 antagonists, and the interaction that we saw at the start of the simulation were also maintained during the post-docking. And also, interestingly, some of the interesting side chains that were interacting with hydrophobic patch were also found in some TAR1 structure, crystal structure, some basically interestingly basic hydrophobic patch found from our model. Coming to my conclusion, I would like to say that we identify 3 new antagonists coming from poor ligand, but also receptor information. We provide also a comprehensive structural analysis of the TAR5 binding site, and also all this work has been published, if you are interested, in the Journal of Chemical Information and Modeling last September. And also this workflow of refinement, the binding site, can be also applied now to other AlphaFold model, but also to other GPCR that we want to find novel ligands. And also we are working on the heat optimization of these agonist and antagonist. And also 2 days ago, we received nice news that we found 2 additional antagonists that we can use to model and improve our understanding of the binding site. And I want to remember that olfactory GPCR are also basically GPCR after all. Saying so, I want to thank you, my supervisor, Antonella Verena, for which I share part of the computational work, Carlotta, Stefano, and Roman for the experiment. I wanted to thank you for the attention. If you're interested, tonight we will have Clarissa, a master student in our lab, that will present a poster for other trace amino-associated receptors. If you want to reach me out in Twitter or by mail, or I'm really happy to take any question now.
Thanks, Alessandro, for the very nice talk. It was amazing. I see that already we have 1 question in the chat, and probably we can take more, I mean, depending on the answer, I assume. So it's from Janelle Lanham and says, wonderful talk. TAARs are a personal favorite of mine. Do you plan to repeat this process with any human TAAR receptor?
Yeah, thank you for the question and for the nice talk. Yes, we want also to target other human TARs since basically they can be interesting drug target, but also because they are still orphan receptors and we know more data to get to know this interesting class of receptor more. But we can also reach by mail to get to know better our project. Thank you.
Thanks. Yes, and I want to remind everyone that tomorrow we will have parallel breakout sessions, like on each of these sessions that we will have for short talks. I see someone, maybe I saw TC turning on camera. Did you have any question? Anyways, if you have any, please follow in the chat and we will continue with our. Thanks again, Alessandro.